mean quiet. I mean quiet. That's what I mean, is the quiet. This is TWIS, This Week in Science, episode number 584, recorded on Wednesday, September 14th, 2016. Are we drunk yet? Hey everyone, I am Dr. Kiki, and welcome to another This Week in Science. Tonight on the show, we're going to fill your head with consequences, viruses, and the moon. But first...
Disclaimer, disclaimer, disclaimer. The wisdom that comes from a life well lived is worth its weight in years. And living life well, as subjective a subject as this may be, must, in this humble opinion, be a life lived in inquiry, an examined existence. Not just in the know thyself sort of way, but in a, the world around you scenario. Because if you don't understand anything beyond your own shadow, if you know nothing of the world beyond your cave walls, what can you claim to have lived? Throw yourself out of yourself. Get up and get out there where the people are, the makers and the doers. They're busy making and doing things, where the investigations are, where the experiments are, where the learning is happening now. Turn on, tune in, open the door and have a look around. See what's out there. Or at the very least, just listen for the sound of This Week in Science coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And good science to you, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk all about the world of science. And it's a big, wild world out there. In fact, it's a big universe of science. Who knew? Who knew? I have some cool stories this week about a potential power crash, some consequences, and a virus inside us.
I've got the Infrared Emperor's New Clothes, Ancient Blue Jeans, How to Paint a Moon, and Tiny Pterosaurs.
And Blair, what's in the animal corner?
Oh, I have giraffes. I have invertebrate sex. Hooray. And I have hibernation.
Hopefully you're not hibernating.
Oh, no.
Because we need you. We need you on this show. No time to sleep. Never enough time. All right, you guys, let's dig into the science news. Big stories this week. The Gaia observing mission looking at the European Space Agency has this program going on. It's a star mapping mission called Gaia. It's a 750 million pound project, which means it's probably around
Heavy, very heavy.
I know, around $1 billion US dollars.
Very heavy.
And this star mapping mission is basically looking at the sky and trying to catalog as many stars as it can see. It just released a new catalog, and it now contains sky positions for 1.1 billion stars. It's a lot of stars, and this is the Milky Way galaxy in greater detail than we've ever seen it before. And 400 million of these 1.1 billion stars have never been seen before, which is pretty cool. Yeah. And so this cataloging goes to show that our galaxy is even bigger than we thought it was. And if our galaxy is even bigger than we thought it was, then probably there's a whole bunch of stuff out there that we haven't seen before. Very likely. For a lot of the stars, according to this article, the positional accuracy of this star mapping mission is 300 micro arc seconds, which is kind of analogous to the width of a human hair if you were looking at it from 30 kilometers away. And so
that would be very hard to see.
Very hard to see, but this telescope is able to do that. So it's going to be running until 2019. A second data release is planned for 2017, and the second release will be hopefully more accurate, up to 10 micro arc seconds, which means that would be like a human hair at a distance of 1,000 kilometers. Yeah, so they're trying to get distance from us of these stars, also the motion of the stars, so that we can get an idea of gravitational influence of what's going on in the Milky Way's gravity field. So according to one of the researchers, knowing stellar distances is extremely important for many fields of astrophysics. And so this is very exciting. Also, future facilities like the Large Synoptic Survey Telescope, the James Webb Space Telescope, and the ESA's Euclid satellite are going to be able to use this Gaia catalog of stars to be able to focus their observations of the future. Very exciting. So anyway, the Milky Way, a little bit denser with stars than we thought it was. Another adding 400 million stars to your immediate neighborhood. That's a pretty big development right there. Absolutely, right? And if we're adding stars, what else is there that we're not seeing, right? Moving on from distant stars to our local star. You know, our star has outbursts every once in a while. We call them solar storms, and occasionally they are aimed in our direction. And depending on how vibrant, how eruptive these solar storms are, they can have different effects on our planet. And you know, one of the great things about the solar wind and the energetic particles that come away from the sun is that we have beautiful auroras on our at our poles, and these auroras, the aurora borealis and the aurora australis, you know, they're gorgeous things to look at, but they are indications of the energy that our planet is being bombarded by. It's a visual cue to how energetic the material coming from the sun to our planet actually is. And sometimes those the aurora in the northern hemisphere will reach pretty far south. It'll reach into Oregon, or even, you know, the mid latitudes of the United States. So people are wondering, all right, we've got these electrical grids on our planet and like in our country, and they're all kind of old and hatched together. And of course, localities have been working on updating them and making them better. And we've reported in the past on increases in solar storms and the possibility that big solar storms could disrupt electrical grids. Well, this is a new story related to this, in that researchers have been going around sticking little sensors in the ground. And you're like, what? Sensors in the ground? We're talking about the aurora. What is going on? Well, they've been using these ground sensors to be able to measure the conductivity of the Earth's crust. And so they're getting a picture. The United States Geological Survey is getting a picture of how electricity flows through the Earth's crust, because normally, rock is not very conductive, and when solar storms come into the planet through our atmosphere, they're very often going to go through the metal grid of the power infrastructure to complete their circuit, because you have the electrical circuit, flow of electrons, and they're going to, if they can't flow through the ground, they're going to find something else to flow through. And that's where the problem with our metal power grid infrastructure comes in. The second thing to this is that this survey that they've done has discovered that there's an area of the United States, the Midwest specifically, which has a very interesting makeup. So the upper Midwest has two types of rock that actually are really good at, when they're together, carrying an electrical current. So they have sedimentary rock, which is a little bit more conductive, underlaid by insulating igneous rock. And so we've got these two layers of rock that, because of the way that they sit together, it creates a dielectric, which is a local electric current that can be kept up during solar storms, geoelectric storms. Yeah, so the idea that comes out of this is that because of its special crustal makeup, the upper Midwest might be prone to a serious power grid disruption should a big solar storm come through, because of the way the ground is all set up to carry currents.
So then is this also, though, I mean, there's a power disruption possible, but is this something we could harness?
Right. That's another
right? You turn it into a big ground-based battery collector, or, so you get the solar storm, and then you've got, I don't know, flywheels, batteries on the ready, something that can sort of capture this energy as it transfers through the crust. Interesting.
Yeah, it's a pretty fascinating finding. And of course, they haven't surveyed all of the United States yet. So they've looked at an area of the Pacific Northwest into kind of the Rocky Mountain area of the United States. So it entails kind of like a bit of Northern California, about the upper quarter or third of California, into Oregon and Washington, and across to about the Rocky Mountains, Colorado, etc. And then this swath from the upper Midwest down through the Great Lakes and through the Midwest down to Florida and the southern coast around Florida and Georgia. So there's a lot of the United States that has yet to be surveyed, but it's very possible that the conductivity of underlying rocks might affect the power grid in the case of big storms. And so we either, like you said, Justin, have to take advantage of it or figure out how to deal with it when it happens. Yeah. And then my final story, we've talked a bit about synthetic DNA on the show, and we reported on a lab that has created 2 additional bases for DNA. So normally DNA are considered nucleotides. They created what are called nucleosides. They're basically nucleotides, but they don't have a phosphate group attached, and they called them D5-SICS and DNAM. Anyway, these are artificial nucleosides that can combine with other nucleosides and nucleotides to create DNA, an expanded DNA library. And this is a really exciting idea. It's like, oh, can we create new protein structures? Can we create chemicals, molecules with additional functions that we haven't seen in nature before? Can we learn about the functionality of how these nucleotides, nucleosides work together to create the proteins that make us up? Anyway, they went looking at these nucleosides in living cells, and they found that using these nucleosides is a bit of a problem when it comes to life, and specifically life on the surface of Earth. Having these nucleosides incorporated into the DNA of E. coli and other cells makes the cells more susceptible to damage from sunlight and even fluorescent light. So phototoxicity is higher when these nucleosides are involved. And I love the title of their paper, which is in the Journal of the American Chemical Society, Unintended Consequences of Expanding the Genetic Alphabet.
Yeah. So I think the lesson here might be nature found a way, and that's not it.
Yeah, nature found a way, and
there's a reason nature set up the way it is. It's because it's tried everything, and some stuff didn't work.
Right.
And what you're left with is what works.
Right.
And so that might be kind of what we find, is we might be stumbling into and sort of adding what were evolutionary dead ends and discovering why they were evolutionary dead ends to an effect.
Yeah. One of the interesting aspects of their study is they wanted to see how the phototoxicity actually affected living cells. And so they used living cancer cells, epidermoid carcinoma cells. It's a type of human skin cancer. And they treated the cells with the D5-SICS. And they exposed them to a low dose of near visible light, and they just didn't grow very well. It decreased cell proliferation. So it indicated that these cells were photosensitized and had damage. And they think the damage occurs as a result of the production of reactive oxygen species inside the cells, and that these nucleosides, when affected by light, lead to reactive oxygen. Reactive oxygen causes cell damage, as we know. Anyone who tries to eat well to reduce radical oxygen in their body. And the interesting thing, what they're talking about, though, that maybe, even though this is something that doesn't necessarily work for living cells, maybe it can be used to treat cancer. Like maybe there's some way that we can actually treat cancer cells with these nucleosides and then just shine light on them.
Yeah.
And kill cancer. So maybe there's some upshot from this, aside from, this is something we didn't really, expect to happen. Yeah. Which is pretty cool. So anyway, expanding the genetic alphabet, it's not as easy as we hoped. It's going to, back to the drawing board, take more work. All right, this is This Week in Science. Justin, what do you have?
Let's see, where to start this. Okay, so a 6,200-year-old indigo blue fabric was found in Huaca, Peru, making it one of the oldest known cotton textiles in the world, and the oldest known textile decorated with indigo blue. The exotic nature of the color, indigo, so intrigued Europeans at one point that it is falsely believed to be a color of the rainbow. It's in there. They say, it's blue, then indigo. There's no indigo. I don't care how hard you look. There's no indigo in rainbow. Some of the world's most significant technological advancements were developed first in the New World, says Dr. Splitzauer. Many people, however, remain mostly unaware of the important technological contributions made by Native Americans, perhaps because so many of these technologies were replaced by European systems during the conquest. However, the fine fibers and sophisticated dyeing, spinning, and weaving practices developed by ancient South Americans were quickly co-opted by Europeans. So this is discovered in a desert area in the north coast of Peru. The experts believe it was likely a temple where a variety of textiles and other offerings were placed, possibly part of a ritual, don't really know. Development of indigo dye was critical for future trends in fashion, fabrics, textiles, arts, Splitzauer says. The cotton used here in the Huaca Prieta fabrics is Gossypsum barbadense, whatever that is. It's the same species grown today and is called Egyptian cotton. So this is actually an American development. But this is sort of, and it's not only the cotton connection we made in this excavation. They also found other cottons that were being utilized. And Spitzauer says here, we may well not have had blue jeans if it weren't for the ancient South Americans. Both the cotton that we use and the dyeing came from. So very interesting ancient textile story there. And now,
and Levi's, I'm sure Levi's is thanking them profusely. Levi's, the Gap.
Probably not.
I love my blue jeans. Can't imagine without them.
I'm wearing mine now. So this is the one of those new segments, Chicks, where we partially are doing a fake sponsor for a product. So here goes. Tired of wearing the same clothes? Made from fabrics that keep your infrared radiation from radiating, making you hot and irritable? Think of the hours each week spent sweating needlessly. The money you could have saved on air conditioning, the social irritation that could have been avoided, the relationships at home and work improved. Then multiply all that time, money, and energy wasted by the billions of irritable humans on this planet and, well, you see, don't you? Yes, it's time for a change of clothes. But not just any clothes. Nanoporous polyethylene cling wrap clothes. The clothes with nano-size holes. The material lets heat escape instead of trapping it like traditional fabrics, according to Stanford University materials scientist Yi Koi, giving cool clothes a new meaning, a more literal meaning anyway. But the question remains, who would wear cling wrap as clothing? Every person who wears clothes could be a potential user of this product, says MIT physicist Svetlana Borinskaya. Current cooling devices include wearable fans, wicking fabrics. These both rely on evaporation to cool human skin. But our skin also sheds heat in another way, infrared radiation. Clothing holds this heat close to the body, Koi says, and if infrared radiation could instead pass through the fabric, he reasoned, people would feel a lot cooler. But the fabric would have to be transparent only to infrared wavelengths, otherwise we'd be walking around in see-through clothes. So that would be cool, but not necessarily cool.
Not necessarily what people really want, unless, of course, I mean, I don't understand. I don't think nudists would really go for the transparent clothing, but
well, yeah, it's
just
got
all
to
the
be
restriction
naked.
with none of the fun, right?
yeah.
Yeah, I don't know. Yeah, it's sort of like, well, is there a point that you can see other people's nudity, or is it that you just want to be free to the open air? In any case, so
Deep Thoughts by Justin. Yes.
yes. So the fabric would have to be transparent only to infrared wavelengths. To visible light, it would need to be opaque. Otherwise, then, yeah, we have the nudist colony Earth. Koi found just one material that satisfied both requirements, a commercially available plastic used in lithium-ion batteries. The material, called nanoporous polyethylene, or nano-PE, is a cling wrap-like plastic that lets infrared radiation through. But unlike cling wrap, this material is not clear. It blocks visible light. Tiny pores speckled through the fabric act as obstacles to visible light, Borinskaya, the MIT physicist, says. When blue light, for example, hits the pores, it scatters. So do other colors. The light bounces around in different directions and scrambles together. To the human eye, the resulting color is white. The pores scatter visible light because they're both in the same size range. The diameters of the pores span 50 to 1,000 nanometers, and the wavelength of visible light range from 400 to 700 nanometers. Infrared light emitted by the body has a much larger wavelength, 7,000 to 14,000 nanometers, so the plastic's tiny pores can't block it. To infrared light, the pores are barely bumps in the road, not barriers at all. The pores are kind of like small rocks at a beach, Borinskaya says. They'll interfere with the motion of small waves, but big waves will wash right over.
It seems a lot like is this something along the lines of the invisibility cloak technique where it's the scattering of light, but it's scattering, instead of scattering light around something so that you just don't see it, it's scattering light so that you see a white color, so it looks opaque. So it's like the opposite. It's like a see me cloak.
Yeah, they tested this. Yeah, see all of me.
See all of me cloak. Yeah.
So they tested the nano PE. They laid it on a hot plate. They warmed it up to skin temperature, and they did it in Celsius for some reason. 33.5 degrees Celsius. They put this hot plate. Then they put the nano PE on it, and it raised the skin temperature by just 0.8 Celsius, up to 34.3. Then they did the same experiment with cotton, and it rose to 37. That's a whole lot more Celsiuses.
Yeah.
Kind of made it hot in there, right? They also tried some techniques to make it more wearable, because currently it is like basically a cling wrap type material. But they're gonna next focus on a way to weave the fabric to make it feel more like a traditional textile. My take on this, though, is he says within 5 years somebody will be wearing this. Within 10 years, I hope most people will be wearing it. But before you go and invest in nano PE, let me tell you, the smart money on this is invest heavily in any company that's making or working on infrared goggles. Because I think, and if I'm not mistaken, I might be right, I think that's going to be still visible light. You will still be naked in the infrared. So this has that sort of an Emperor's New Clothes feel to it somehow.
Right. So depending on the kinds of glasses or eyewear people are donning, they may see people different ways.
Now, yeah.
It seems,
if you want to, you might not want to. It might actually be rather disturbing to see everybody walking down the street naked.
Well, it would be funny if there was something going on in current glasses. Like I think all the time about my polarized sunglasses, that you try to see a screen and it doesn't work. It'd be pretty ridiculous if those had just been in glasses for a while, and then all of a sudden you're like, oh my God, everyone, put it away.
It seems, though, that, I mean, this is nice. It'll be holding body heat in. So maybe people in Antarctica this will be great for like Antarctic or expedition wear if you're going to the top of a high mountain to hike glaciers where it's cold, or maybe maybe it's long john wear for astronauts. Space is cold. Maybe it'll help.
Space is hot. Space is boiling hot. Space is thousands of degrees.
Oh, right. It's hot, but there's no molecules. No molecules to move the heat, transfer heat away. But so you're cold. Yeah.
But it's thousands of degrees hot. It's so complicated. I don't understand space. Outside of the spaceship, it's super hot, but there's nothing about touching it to transfer the heat around, so it just doesn't.
So it's cold, so you freeze.
But it's hot.
It's
2,000 degrees, and you freeze to death.
Either way, this nano PE could be great naked wear for the space station.
But yeah, so it's, I mean, it's got some really interesting potential.
Yeah.
But they have to make it a clothing that feel. I mean, it's one of those innovations. Like, yeah, we've developed this idea for plastic clothes, but if they don't feel comfortable, if they don't get the ergonomics of somebody wanting to wear them down, then they'll be no good. But we wear plastic all the time.
We do. Fleece.
We do.
Mm-hmm.
Yep. Polyesters.
Mm-hmm.
Micro polyester something.
Mm-hmm.
There's another one. Yeah.
That's right. Plastic for the people. Put it on, people. Put it on. This is This Week in Science. You know what time it is here at TWIS? It's time for this week's Blair's Animal Corner. Maybe that's what time it is.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels that are no closer.
What you got, Blair?
Oh, well, we're going to start with something that's all the rage on the internet this week. I want to talk about giraffes.
I like
So-
giraffes.
Yes. The internet is
We
abuzz.
have different internet feeds, you and I.
Yeah.
Let me just say.
My internet, at least, is abuzz with the giraffe news, and I want to talk about it. So, historically, scientists have recognized a single species of giraffe made up of several subspecies. Depending on who you ask, it's 6, 7, 8, or 9 subspecies of giraffe. But so all of them had the same genus and species. But now, a recent study on giraffe genetics says there are actually at least 4 distinct species. So previously, all of the giraffes were under Giraffa camelopardalis, and now there are these 4 species. The southern giraffe, Giraffa. See if you can remember that one.
Giraffa.
Yeah,
I love it.
The Maasai giraffe, Giraffa tipilskirchi, the reticulated giraffe, Giraffa reticulata, and the northern giraffe, which is still Giraffa camelopardalis. So now identifying 4 distinct species changes things in a few ways. But first I want to talk about how this happened. So giraffes, they all live in Africa. They all look pretty similar. They all have a pretty similar diet. They all have a pretty similar life history. The only thing that you can really tell about them that's different is that their patterns are slightly different. If you're not someone who studies giraffes or works with giraffes, it's pretty hard to tell the difference in these subspecies or what were considered subspecies based on their pattern. The other differences are their range, their home range, and most of these subspecies do not interbreed with each other, despite human pressures on their habitat. But that coupled with the fact that they are very understudied in terms of large mammals. They're a group of large mammals that people don't do a lot of scientific studies on. Elephants, rhinoceroses, gorillas, lions, those are all African mammals that get way more research done on them than giraffes.
Why do you think that is? I mean,
My personal opinion.
Every zoo has giraffes.
Yeah. My personal opinion is that they're not that, what I would call, these are giant air quotes, sexy, right? They're not a carnivore. They're not considered to be super smart. They're not, they don't have complicated social structures. They are in many ways just a stretched, hooved mammal. And there are a lot of hooved mammals on this planet. And unless hooved mammals are your thing, I think that the average person, maybe they don't care as much about hooved mammals. Giraffes are basically stretched cows. So I think that they are underutilized in terms of their research potential. Now, based on all of this, despite the lack of research, they've actually been dropping pretty significantly in their numbers. They're over the past 3 decades, so 30 years ago, it was estimated there were about 150,000 giraffes in the world, and now there's less than 100,000. That's a pretty big dip. And it's because of many things, but it's mostly because of the number one cause of animal numbers to drop, which is
habitat loss.
Habitat loss, exactly. So that's the main concern for giraffes. The big caveat here is that if you're looking at listing animals as endangered and using governmental or conservation funding and or political power to protect a species, it has to be a species, not a subspecies. There's not a lot out there to protect subspecies. And specifically, the IUCN, which lists animals as endangered or threatened, they only list species. So if you're looking at giraffes, the number of giraffes in the world are not low enough to list them as endangered. If you separate out these individual new species, there are some that are actually potentially critically endangered. And that's where this becomes really important.
Yeah, so are people going to start debating the potential bias in separating these species. I mean, it's genetic tests. You show that they're separate species or they don't breed together. Okay, they're separate species, but they're still all very closely related.
Sure. For comparison, the genetic differences between these 4 giraffe species are at least as great as those between polar bears and grizzly bears. So genetically, they are vastly different. And it's because they have not been interbreeding for a really long time. So first question I would have is why? Why aren't these subspecies interbreeding at all?
Because they don't have the right spots.
Right.
Yeah.
I don't like that guy. It looks less like a net and it looks more like splotches.
Yeah, you're all splotchy. And there's different colorations to these markings as well. Right? Some are lighter versus darker. But are the sizes of the giraffes? I mean, I think these vary a bit too.
They're pretty comparable, the size and body shape of these animals. There's nothing physically stopping them from reproducing with each other. It's all clearly just that they I would assume can recognize each other as separate species.
Interesting.
But yeah, it's there have been lots of debates about this. The main thing I would just offer up to consider, which I'm not saying a specific opinion or what's right or what's wrong, is that the reason this study is so important is that we can't list subspecies and protect subspecies the same way that we can protect
species.
But I would argue that individual subspecies have just as big of an impact on a system and an environment as a potential species.
Yeah, and you have argued that before. We've had that conversation many times.
Yeah.
Yeah.
And so the
what we
need
need
to make
to
this
do
distinction is kind of like it's just
we should be saving systems. We shouldn't be worried about these species so much. But if we are going to be worried about the species, then what we need to do is have some documentaries where there are lions chasing giraffes or hyenas taking down giraffes so that we see the threat to the giraffes. Because in documentaries and at zoos, they're just sitting there chewing their cud, chewing on those tree leaves.
Yes. Well, as much
giraffes.
As much as it's predation at the cost of habitat loss, it's also a big part of it is you have to remember that these animals are herd animals and they have one male and many females. And that means they need huge expanses of land because they're also browsers. So they need this huge amount of land to just be able to munch on leaves all day. Yeah. And then on top of that, they need to have space for full-sized herds of many females and one male. You need a bunch of those. You can't have just 1 or 2 herds because then you lack genetic diversity, and we know what happens then. So within what was a subspecies is now a species, you need enough space to have all these different herds because at a certain point, that one head male is fathering every single baby for at least a couple years in that herd. So if you're going to have any genetic diversity at all, you need space for bachelor herds, you need space for several full-sized adult herds.
And Ed from Kinetic asks, Blair Baz, do these different giraffe species mate in zoos? And it's sort of interesting because I believe with all the giraffes that are at the Sacramento Zoo, the baby came from 2 that were the same type, even though there's multiple types kept together.
Yeah, I haven't really heard much about them. They happen occasionally, but to my understanding, it doesn't happen very often. But that also is because, at least in America, in zoos, there would be an effort to keep those, especially because some what were subspecies and now species of giraffe are so few. So, for example, northern giraffe, the last one that I said, Camelopardalis, is less than 4,750 in the wild. And the reticulated giraffe, reticulata, less than 8,700 individuals. So that's a very low number. So if you're looking at zoos like seed vaults, like we've talked about before, then
Right.
you don't want to mess that up. Even if they were only subspecies at the time, you still don't want to kind of confuse those lines, even in zoos, because if you're going to release those animals later, you need to have a pure line, especially if they're adapted to different habitats, even slightly.
That is right. What's up next in the animal corner?
Oh, yes, I just have a little dash of invertebrate sex. So looking at our good friend, the roundworm, or as many researchers call it, the worm, C. elegans. They are a great animal to study for a lot of different things, particularly when it comes to chemical exchange. They're the transparent roundworm. They live in kind of a fluid mass, so it's very easy to kind of see what their response to different chemical changes would be. This specific study coming out of University of Chicago was looking at chemical cues from males to females when it comes to reproduction. The long and short of this study was that chemical cues from males can make women or females go into puberty sooner, maintain reproductive age longer, but as a trade, greatly reduces longevity. So
Okay, so you can reproduce longer, but you're not going to live as long. So all your
Right.
energy is going into reproduction.
Right. So they used C. elegans. They identified two distinct signals produced by males that affect female reproduction, and the females then directly altered their physiology. And they don't see this as an intentional side effect. It's more like a cost benefit. So they see it as kind of something that is a drawback. They called it collateral damage to increasing reproductive age on both ends of the spectrum for the females. So from this, they were able to kind of parse out where this was coming from, and future studies in this vein, they believe, could help improve fertility science for humans, because actually all of these chemical cues are pretty much the same in most animal species. So they think that this could help with fertility research. They think also they could identify the part of this chemical signal that is related to reducing life expectancy, and they could use that also to prevent that from influencing humans as well. So based on this pretty small study, there's some pretty lofty goals coming out of this.
Yeah, I mean, we use C. elegans as a model for so many aspects of medicine and understanding our own genetics and physiology. I mean, it is a simple organism, but there are so many aspects of this simple organism that are highly conserved, even in humans. And so it's an interesting question, as we mess with our reproductive systems, as we take fertility hormones at older ages to have children, what effect is that going to have downstream to female human longevity? We don't know the answer to that question. So I find it fascinating. And maybe even, what about women who take the pill and kind of delay their reproduction? How does that affect it? So there's some interesting questions that are tied up in reproduction and longevity. Who knows if they are tied up in the same way in humans as they are in these worms?
Yeah, absolutely. And that's such a good point with the birth control pill too, because in this study, these are tiny animals. The male didn't even have to be there to present these causes or these changes in the female. They just needed a teeny tiny amount of the male pheromones, and that affected aging in those females. And what's really the reason they kind of thought this could jump to humans is because the results they got here echoed previous findings in mice, which means if it can jump from the roundworm to the mouse, it's a much shorter jump from the mouse to a human.
Yeah.
Yeah, if you're looking
Theoretically.
at the amount of genes you have in common, right? It's
Right.
pretty similar. So also the, just the reproductive system of a roundworm versus a mouse and versus a human. It's a much shorter leap to take.
Absolutely.
Yeah, it's pretty interesting.
All right, everybody. I think it's time for us to take a quick break. We'll be back in just a few more minutes with viruses, moons, and tiny pterosaurs. And then we're going to talk about getting drunk. That's up next on This Week in Science.
Don't miss it, man. It's going to get power. We're going to have the kicks. Everything's going to be awesome, man.
Hey, everybody. Thanks for watching or listening to This Week in Science. Your patronage really, really is what keeps us going. It's the reason we go on. Well, I mean, we love the science and we love talking about it, but it's all about you. You are the people who keep this little tiny show afloat. You are the people who keep us doing this week after week after week. I want to tell everybody I went to the Maker Faire this last weekend and had a little booth. There are videos available at our YouTube channel, This Week in Science, on YouTube, of interviews that I did with a couple of scientists and makers at the Portland Mini Maker Faire. So you can go check those out if you are interested in seeing a little bit other content, things that we're doing in the world, who's making in Portland. I talked to a few of those people. And there's also an iTunes podcast feed that Paul Colligan put together for the audio files of these interviews, along with his interviews, and that is at iTunes. It is the podcast Maker Faire. It is the Portland Maker Faire podcast. So you can look for that feed on iTunes and hopefully enjoy it. TWIS also has merchandise that you might enjoy. We have t-shirts, and you can head over to twis.org to get some of those t-shirts because we have a link on our website that goes to our Zazzle store, and that Zazzle link has all of the t-shirts, sweatshirts, hats, cups, mugs, phone covers, all the things that you would love to have with the TWIS logo emblazoned on it or some of Blair's original artwork. TWIS is also supported by listeners like you. You are our producers. Your donations pay for our hosting, bandwidth, contractors we need to hire, fun things we try to do for the show from time to time. And we really appreciate any amount that you're able to give, whether it's $1 or $100. Every penny counts. It really helps keep things flowing and moving here at TWIS. So whether you're an old listener or a new listener, or you're watching on YouTube, please consider supporting us. And you can do that a couple of ways. Through Patreon is one. We can go to patreon.com/thisweekinscience. There is a link in the main header bar at twis.org to be able to go there. The other way is through PayPal donations. Again, there's a link at twis.org for PayPal. Makes it quick, easy. You can make it recurring or one time. You can figure out how you would like to be a part of supporting and producing TWIS on a week-by-week basis. If you can't afford a donation, there are many other ways that you can help us out. We can always use your help on, there are always things I'm looking for getting done for the show. Totally. But biggest thing is get your help telling people about TWIS. Go to a Maker Faire, tell makers, scientists you meet on the street, because everybody should meet scientists on the street. Tell them about TWIS. Tell people who are just cool cafe cats about TWIS, because maybe their earbuds, whether or not they are connected by wires to their phone, should be filled with the tunes, the dulcet tones of This Week in Science's voices. However you are able to help, we really, really thank you for your help. Thank you for your support. We could not do this without you.
Your chakra and chi are both growing real strong. Your cold disappeared after just 9 short days, all thanks to the words on the whole earth displays. Now due to the juices and pills and the creams, the body's lost toxins, whatever that means. You've stopped eating all of that sinister food. Your dinner tastes awful, so it's gotta be good. And still, you can't believe what a skeptic I am. I can't believe you believe in that shell. We disagree, but I still give a damn. The ramifications.
And we are back with more This Week in Science. I've got a story about a virus inside us.
Uh-oh.
Are you ready for this?
I'm scared.
Don't be scared. It's a virus that's fantastic. It's an old virus that somehow a long time ago became integrated into our DNA. And it is the virus that gives us a protein that enables us mammals to form a placenta. Syncytin is the name of the protein that is formed from this little virus's DNA that is integrated in our own. And so it's fantastic that we have it. We wouldn't really be mammals without it, right? Yeah. So this group, this group from the University of Copenhagen published a study in Nature. Copenhagen. They published a study in Nature this last week, and they're like, well, does this syncytin influence anything else? Is it only involved in the placental formation? Because in viruses, what it does is it causes membrane fusion, so that you get the host cells to fuse with another cell and then empty a viral load of DNA into the cytoplasm of another cell that it merges with. And so the syncytin in mammals in forming the placenta has a role in cellular fusion that allows the formation of the outer layer of the placenta. We acquired it about 30 million years ago. It's fantastic. So there are a couple of forms of syncytin, and these researchers, another group from the Centre of France's National Centre for Scientific Research at Université Paris Sud, they deleted a version of syncytin called syncytin B from mice. They said, okay, let's just take that out of the mouse genome. And so they're creating what's called a knockout mouse. They found that, okay, female placentas weren't forming very well in the females, but male mice weighed less than animals that had all their syncytin. And so they're like, we didn't really, maybe syncytin has something to do with males. We didn't think so before because they'd only ever really seen it in the placenta. And so this syncytin, other previous researchers found that it is active in immature muscle cells. And mature muscle cells form through fusion of these immature muscle cells. So they found that the muscle mass was the reason for the weight difference in the males. And so this discovery finds that not only is syncytin essential for fusion of cells for the outer layer of the placenta in females, it is a major player in the larger muscle mass production that males tend to form compared to females.
So this is 30 million year old coevolution with this virus.
Yes. This virus, this viral protein that ended up in our DNA helps us a lot. It makes males more muscular. Females lets us have babies. That's fantastic.
Thank you, ancient virus protein remnant in our DNA.
Yes.
Another piece of proof that we're just a bundle of bacteria and viruses with some protein.
I know. The virus is inside us.
Well, I think it also illustrates that one of those things that we tend to think of only happening on the level of bacteria, which is that sort of information exchange of DNA taking place. Like it only happens at that sort of really primitive level. But obviously, this incorporation, at least by viruses, into our DNA has allowed the human evolution to take advantage of things that could be useful.
Yeah, so these syncytin proteins are probably in all sorts of other, they probably function in all sorts of other areas. We just haven't figured out what they are yet. And then there are other retroviruses and their remnants that are in our genome that we have no idea about. I mean, there's so much inside of us. We haven't, it comes from other places that we have no idea. It's good.
Yeah, and with this discovery, look for a really, really strong, fast Russian Olympic running team.
Right. That's right.
Some 20 years from now.
Exactly. Hey, Justin, you have a story?
I do. I've got a few of them, actually. What do you want to hear about first? How about we talk about the New Horizons spacecraft? The
Yay!
large red dot. No, we're not talking about Jupiter anymore. This is a large reddish polar region on Pluto's largest moon, Charon. It looks like it has been spray painted on. And it was puzzling scientists who had no explanation for it. They could have assumed aliens did it and given any number of reasons for its meaning, like calling it a warning of imminent destruction of Earth if research funding isn't significantly increased. But they didn't. Instead, they looked into how it got there. And they think they have solved the mystery. Charon's polar coloring comes from Pluto, as methane gas that escapes from Pluto's atmosphere and becomes trapped by the moon's gravity and freezes to the cold, icy surface at Charon's pole. This is followed by a chemical processing by ultraviolet light from the sun that transforms the methane into heavier hydrocarbons and eventually into reddish organic material called tholins. Who would have thought that Pluto is a graffiti artist? Spray painting its companion with a reddish stain that covers an area the size of New Mexico, asked Will Grundy, New Horizons co-investigator from Lowell Observatory in Flagstaff, Arizona, and lead author of the paper. Every time we explore, we find surprises. Nature is amazingly inventive in using the basic laws of physics and chemistry to create spectacular landscapes. So that's pretty cool, right? This is the team combined analysis, detailed Charon images obtained by New Horizons with computer models of how ice evolves on Charon's poles. Mission scientists had previously speculated that methane from Pluto's atmosphere was trapped in Charon's North Pole and slowly converted into the reddish material. But until this study, they had no models to support that theory. But this model says, you're right. That's how it happened.
Awesome. And I also love some of the names of these areas. The North Polar region is named Mordor Macula.
Mordor.
And it's red. The reddish Mordor.
What a bunch of nerds.
Totally.
Yes.
Oh my goodness. Moving on from Charon and Pluto to closer to home. How about the moon? We like the moon.
The moon.
We like the moon because it's close to us. We like the moon, but not as much as a spoon.
Huh? Don't understand that you say.
Fantastic
But I
song.
love the moon. Ah, the moon.
As the spoons are for eating soup and maybe other vegetables. So anyway, La Lune, the moon.
La lune, the moon.
There have been hypotheses for a while that the strain of the tides caused by the pull of the moon could actually increase the magnitude of earthquake tremors. There are many there have been hypotheses related to this for a long time, and there are questions also related to, as sea levels rise, are the tides changing and putting different pressures on the plates in a way that will change earthquakes? So,
Because it's right. I mean, you look at how big the ocean is and the mass of the water in there, and if
that's the water.
Yeah, it's a lot of mass built up in there, and if the moon is affecting tides globally, it's sort of shifting that mass about on our surface, and you could infer that there should be some sort of influence on something. Maybe not as to the degree of plate tectonics, but maybe.
Right, not to the degree of plate tectonics, and it probably one of many influences into the magnitude of earthquakes. But there's a study published in Nature Geoscience this week from a seismologist at the University of Tokyo named Satoshi Ide, and he and his colleagues investigated earthquake records in Japan, California, and the entire globe. And they looked at what happened to relative tidal stresses for 15 days leading up to the occurrence of every quake that they looked at. And they found, they assigned a number to the tidal stresses, and 15 was the highest amount of stress. And they found that some of the large quakes occurred pretty much at the time of the highest amount of strain due to the tides. And so this was during new and full moons. And this is the reason that the maximum tides occur at new and full moons is because it's not just the moon that's pulling on the mass of water. It's a lining up of the sun and the moon and the earth, so that it's the sun's gravitational pull, the moon's gravitational pull, all affecting the earth. And so they looked at like 10,000 earthquakes, and they found if it was during one of these new or full moon periods, that the earthquakes were more likely to grow from a 5.5 to over a magnitude 8.
Whoa.
Yeah, so
magnitudes we're talking.
Magnitudes we're talking, which is an exponential scale, not just a logarithmic one. And so
yeah, because and this is side note, right? But it's like a 5.5 is half the size of a 5.6, which is half the size of a 5.7, which is half the size of a 5.8. Like this is right. This when you start talking about the difference between 6.5, 7, let alone an 8. You're talking about completely different events in terms of the energy that's being released by these tectonic movements.
Yeah, so there are seismologists around who think the results are fairly plausible compared to other hypotheses about how the moon could affect earthquake strength. But like I said before, this is one of several factors that can influence what happens during a tremor and actually gets the ground moving. And so the tides are definitely not going to be the last word on things, but maybe it's another piece of information if areas that are earthquake prone, they can take advantage of this information to plan ahead during certain periods of time, compared, kind of prepare during certain periods versus others. And so now they're doing additional surveys to look at other plates, other earthquakes where the crust of the earth plunges under the continental crust. So see if there's a pattern there also. Looking at lots of earthquakes, checking it out, but there seems to be a link between the pull of the moon and earthquake strength. Mm-hmm. Did you have a mooning story? Or that, I guess Sharon was your mooning story.
That Sharon was the mooning story.
What else do you have?
All I've got left here is a bit of the Late Cretaceous, which
let's hear it.
You and I may not remember too well because it happened a really long time ago.
Yeah.
But when you think of a pterosaur, what do you think of?
Pterodactyls.
Right.
Pterodactyls.
And
it's a
they're big.
pterodactyl. Caw, caw.
They found
in Jurassic Park, what was it, 3?
Yeah,
the wing, and they had the wings.
Wingspans of 30 feet. We're talking like school bus, right? Just huge.
Yeah, huge.
So this is our picture of it. And until recently, there have been a couple that were in the about 8 to 10 foot range, were the smallest they'd found. But this fragmentary fossil they just discovered, roughly 77 million years old, found in British Columbia. This is a Canadian pterosaur. Okay, had a wingspan of just, what would that be? 1.5 meters. That's 4 feet, 5 feet.
Yeah,
4.5, 5 feet.
6 feet.
It's pretty close, they say, to that of a bald eagle.
That's adorable. I want to put bracelets and jesses on that and wear it on my arm.
Right? Can you imagine this at the zoo? Like, this would be a zooable dinosaur, right? Little pterosaur sitting on your sleeve. And this is blah, 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 and come on, you can touch it, but just with a closed hand. I don't want any fingers out there. There we go.
I love this. A zooable dinosaur. That is a new
zooable dinosaur.
It's a new category of dinosaur.
I'm excited.
So cute.
Look at it next to the cat. I want that on my wall.
Cat wants to eat the pterosaur for dinner. This is an actual figure from the study discussion to show the size. We've got an image of a profile of a black cat looking at the profile of this tiny pterosaur, and it's pretty cute. Pretty cute.
Mm-hmm.
Kitty cats and pterosaurs. For the win.
Oh, I love it.
Oh, kitty cats and pterosaurs. Yeah. Tiny, that's cool.
That's
I'd
great.
like to see them.
Totally
so let's
change paradigm.
maybe let's not bring back mammoths. Let's bring back tiny pterosaurs.
Oh, we'll bring that back too. If I ever become, by accident somehow, a billionaire, if I were a billionaire, I would attempt Jurassic Park. I definitely would. I would.
I'm not surprised at all.
I would make that happen.
No question.
I might stay away from velociraptors. I think lessons can be learned from sci-fi.
But now we know the T-Rex is a scavenger, so they're not going to hunt. So they're
unless there's nothing to scavenge. It's one of those things. Like, I think they would adapt to a different scenario if need be. But yeah, I think a T-Rex probably would be fine in a proper enclosure.
Right.
Paleo Dude wants us to bring a Quetzalcoatlus instead.
There we go, Quetzalcoatlus.
Like a pterosaur that's the size of a bus.
Right. Yeah, so that one might be a bit harder to just build the enclosure. But you know what? If I'm a billionaire, what's a
You've got an island. Cage? Just put it on an island.
It's a bigger bird cage. But no, like this would be a very manageable zoo dinosaur. And I would also definitely bring back all of the megafauna. The giant sloth, the mastodon,
the giant ground sloth. Yeah, that'd be great.
Wouldn't it?
Giant
That would
lemur,
be very cool.
please. Giant lemur would be great.
Okay, giant lemur, or the 8 foot tall penguins.
Yeah.
Right? Yeah, those we definitely need to get in there.
Elephant bird.
Yeah. Hey, Blair, you were segueing with giant lemurs, but you've got a story about dwarf lemurs.
I do.
So let's talk about that.
And it's so fun. So hibernation, it's not sleep, despite what you might think and what cartoons might tell you. Hibernation is a lowering of your metabolism and a lowering of your body temperature, depending on how you define it. Bears don't hibernate. They go through winter sleep. because they don't lower their body temperature very much at all. Most true hibernators lower their body temperature as much as 10 degrees Celsius. That is a pretty intense drop in body temperature. Dwarf lemurs, I'm going to attempt to say the genus and species, Chirogalius medius. It's the CH in the front. They're in Madagascar, of course, because that's where lemurs live. And they are the first ever to be found to sleep while in hibernation. They found this out
Wait.
Yeah.
They sleep while they're hibernating.
Yes.
So they're reducing their metabolism and hibernating, and then they're like, I think I'm going to take a nap. Now I'm going to wake up. I'm still hibernating.
And they found this by monitoring their blood pressure, by monitoring their body temperature, their breathing rate, their metabolic rate. But then they also monitored brain waves and eye movement so that they could test for rapid eye movement, for REM. And they noted that it most typically occurred when the ambient temperature was above 30 degrees Celsius. 30 degrees Celsius is pretty warm, and we usually think about animals hibernating in the cold, but these guys hibernate in the warmth, this is actually the species that I talked about just a few weeks ago that could wake up from hibernation when they smelled burning. Remember this?
Mm-hmm.
So these guys are this very same.
Burning.
Yeah. Am I having a stroke, or is the forest on fire? Yeah, and so these are the very same guys that they're also unusual because they hibernate due to drought, not due to cold temperatures.
That's interesting.
So
That's
all
smart.
of this makes them unusual, but it also is very unusual but interesting because they sleep while hibernating. So this is something that we might have to look closer for in other species, or it's possible the dwarf lemur is just unusual in this ability. But still a pretty cool find.
So
Yeah.
wait, so
And it
most
could tell us more.
that hibernate aren't sleeping?
Right.
Like that's what I picture.
No.
They're just
Hibernation is not sleep. You lose all muscle control.
Huh.
Yeah. You're completely immobilized. It's almost like people who go through sleep paralysis. It's more like
So
that.
can you still have dreams while you hibernate?
No, because
No?
you're not having rapid eye movement.
Oh, wow.
Your body
the
temperature
bear-
lowers several degrees Celsius. Your metabolism slows down so that you have maybe a quarter of the heart rate that you had. So there's not enough blood circulating through your body to be able to have your brain doing much.
So the bear, I always
Bears
thought
don't
they're just, like,
hibernate.
taking long.
They sleep, and that's why they wake up, and they can be cranky bears if they wake up early.
Yes, bears go through winter sleep.
I was having a good dream and you woke me up.
Bears just sleep for a long time because there's not a lot of food. But their metabolism and body temperature don't drop enough for it to be considered hibernation.
So next time somebody says a bear is the hibernating bear, I go, no, stop. Bears not.
I mean, if you want to sound kind of like a snooty jerk, yeah. I give you that power.
Okay, thank you, because that's what I was coming for. Actually, technically,
It's like saying it's not called hibernate, it's called
Just take a long nap. That's really what's going on. Hibernation carries a low heart rate, no REM sleep, where the bears can dream, man. Don't you understand anything about the world?
Think about the world. How drunk are you guys? Do you know?
Getting there, working on it.
I'm a 0.
I'm a 0 too. But you know what? According to
That would make me a 0.
according to a recent paper in BMC Public Health, a bunch of researchers went to bars and clubs in Cardiff, Wales, which is a fairly bar and club heavy area that they went to, and they spent their evenings between 8 p.m. and 3 a.m. on Friday and Saturday nights. They went to a bunch of neighborhoods, and every 7th person they ran into or saw, they asked them if they would take a survey. And then they asked people these questions. How drunk are you right now? On a scale of 1, totally sober, to 10, completely drunk. 2, how extreme has your drinking been tonight? On a scale of 1, not at all, to 10, completely extreme. 3, if you drank as much as you have tonight every week, how likely is it that you will damage your health in the next 15 years? So 1, definitely will not, to 10, definitely will scale. 4, and finally, if you drank as much as you have tonight every week, how likely is it that you will get cirrhosis of the liver in the next 15 years?
Way to kill a buzz.
On a 1, definitely will not, to 10, definitely will scale. And basically, the turnout of this study is that drunk people have absolutely no idea how drunk they are.
Shock. Shock and awe.
Mhm.
But the interesting thing that came out of this is that drunk people judge their drunkenness and the health consequences of their drunkenness, of their drinking, based on the social environments that they're in. And so they ended up doing a statistical analysis, and they found that, I quote, "It appears that drinkers are more self-aware of their own level of intoxication when in the presence of those who are sober. And they will judge their drunkenness based on the least drunk person in a group."
So, Justin, are you like an 8? Because
So-
you're with 2 sober people right now.
Wait, oh, so interesting. So they would say, I thought they would go the other way. I figured you would say, well, nobody else seems to be swaying about or staggering, so I must be in good company, so I must be as drunk as they are.
No, I
Or
think about
sober
that.
as
Like,
they-
have you ever run into someone from work that you weren't expecting to see when you've been out and had a couple, and then
Like Karen?
like Karen,
Karen.
and then,
Karen, why are you always showing up at my favorite bar?
yeah,
Karen.
and then you realize you're like, am I slurring my words a little bit? Did I use that word wrong? I feel like, don't you get super self-conscious when you run into someone that you weren't expecting to see who is sober, if you've had even one drink?
Well, yeah, I think I would if it were like 2:00 in the afternoon. I think that I would be like, oh, I should try to
Yeah.
really maintain. But I think if I'm like out at a bar, I don't think I would be like, oh, hey, what's going on? Yo, we're at a bar. What are you doing? Oh, yeah, I know this guy from work. I work with this guy. He's totally a work guy. Yeah, come on, I'll get you a drink. Come on. Like, I think if you've been drinking and you're in a social drinking atmosphere, I don't think you should care that much
And
about
so
the etiquette of
you
the
don't-
workplace.
you don't think, yeah, but people, based on their finding, basically people judge how drunk they are based on the people that they're hanging out with, their peer group. And
Yeah.
as Annalie Newitz puts in this Ars Technica article, if everyone around a drunk person were blackout drunk, an extremely inebriated person would consider himself relatively sober.
Mhm.
And a tipsy person with sober people would be very conscious of being drunk.
Right.
Like like Blair was just commenting on.
I wonder if it's cultural, if there's a cultural bias here. So
Could
this
be.
was done in Great Britain, where maybe people are very focused on being polite, and
Where
maybe
Scotland.
in certain areas where there's a certain social side to being the drunkest person at the bar.
Chico State.
Right. And so from this study, because this is a group working on trying to figure out how to improve public health, they think that maybe it would behoove bars to kind of just have token sober people. Instead of having the sober driver, you just have the sober person at the bar.
That's the bartender, isn't it?
That's the
Right.
bartender.
Right? Yeah.
But wouldn't that hurt sales at the bar?
That was my first thought as well. So it's not necessarily in the bar's best interest.
Well, a lot of this self-reporting too. It's got to be sort of it's self-reporting. It's the environment they're in. I know people who If they've gone out for an evening and had 3 drinks, that was the extreme end of the drunkest that they've been. And I have other consorts that I would, that I hang out with that once you've gotten through your first 6-pack, now you're starting to drink, right? Like it's kind of like depends on the environment of people you're hanging out with, how you would judge what is a lot or not a lot of drinking.
That's exactly what this is saying. It's the people.
Makes sense.
It's the people you're with. Yeah. The people you're with, how you judge it. And I would like to thank the people I'm with for another great show. Thanks, you guys. This is a good show.
So happy to be back.
Yeah, Blair, glad you're back safely from your zoo adventures in Southern California. I saw your social media feeds. They looked fantastic. And I'd also like to say thank you to all of our Patreon sponsors. Thank you to Chris Clark, Paul Disney, Dave Friedel, John Ratnaswamy, Richard Onomus, Byron Lee, EO, Jared Lysette, Ulysses Adkins, Kevin Parachan, Andy, Keith Corsell, Jake Jones, Eric Schwalb, Bob Calder, Mark Massaros, Ed Dyer, Trainer84, Brian Hedrick, Cassie Lester, Layla, Marshall Clark, Charlene Henry, Don Kamarachka, Larry Garcia, Randy Mazuka, Tony Steele, Steve DeBell, Haroon Sarang, Melissa Mosley, Alex Wilson, Jason Schneiderman, Rudy Garcia, Greg Guthman, Dave Neighbor, Jason Dozier, Matthew Litwin, Eric Knapp, Jason Roberts, Craig Landon, Daryl Lambert, Rodney, David Wiley, Robert Aston, Cosmic Gypsy, Brian Hone, Orly Radio, Brian Condren, Pixelfly, Marked, Nathan Greco, Hexator, Deborah Smith, Mitch Neves, Flying Out, John Crocker, Richard Porter, Christopher Dreyer, Ben Rothick, Sylvan Westby, Artyom Shu Wada, Dave Wilkinson, Steve Mishinsky, Rick Ramus, Gary Swinsberg, Phil Nadeau, Braxton Howard, Salgood Sam, Matt Sutter, Emma Grenier, Philip Shane, James Dobson, Kurt Larson, Stefan Insom, Michael George, Russell Jensen, Mountain Sloth, Jim Drapeau, Tara Payne, John Maloney, Jason Olds, James Paul West, Alec Doty, Luma Lama, Joe Wheeler, Dougal Campbell, Craig Porter, Adam Mishkan, Aaron Luthen, Marjorie Paul Stanton, David Simmerly, Tyler Harrison, and Columbo Ahmed. Thank you for all of your support on Patreon. If you're interested in supporting us, you can find more information at patreon.com/thisweekinscience. And remember, you can help us out just by telling your friends about TWIS. So maybe tell 3 people about TWIS this week. That could help us out a lot. And on next week's show, once again, we're going to be broadcasting live online at 8 p.m. Pacific time on twis.org/live. And you can watch and join our chat room there. There's so much fun going on in the chat room all the time. But don't worry, if you don't make it, you can find our past episodes at twis.org/YouTube or just twis.org.
Thank you for enjoying the show. TWIS is, of course, also available as a podcast. Just Google This Week in Science in your iTunes directory, or if you have one of those mobile-type devices, you can look for TWIS, the number 4, Droid app in the Android Marketplace, or for This Week in Science in anything Apple Marketplacey.
And for more information on anything that you've heard here today, you'll be able to find some show notes on our website. Yeah, that's www.twis.org. There you can also make other comments and start conversations with the hosts or other listeners.
Yeah, or you can just contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you have learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. and all it'll cost you is a couple of grand. 'Cause this week's science is coming your way, so everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way, so everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you learned anything from the words that we said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
And once again, we have come to the end of another This Week in Science episode. We hope you have enjoyed your tour of science for the week, and that you will go into the following week more sciencey in your outlook. Welcome to the post show. I have a code in my node. It's in the node, it's a code, and it sniffles.
Oh no.
I made it through the episode. Yes. I still have to finish a presentation I'm giving tomorrow. No. Yay, back to school. I thought I was doing really well. It's getting worse.
Oh no.
Tomorrow's going to be fun. I have to come across as happy, perky, good presenter. Try not to have bloodshot eyes and a sniffly nose, and we'll figure it all out when I get there. Figure it out. Always figure things out. I can do that. I can figure it out. How was your zoo trip?
It was great. So it was the annual meeting of the Association of Zoos and Aquariums.
Yeah.
Which this was my first time going, so that was really great. And it was in San Diego, so I got to check out the San Diego Zoo, and I went to SeaWorld too, so I'll be sure to mention that while Justin's not here. And
right.
I also went to the Birch Aquarium, which was awesome.
Sweet.
Yeah, and let me think, what were my big takeaways? So I learned about this amazing study. Do I have it out somewhere? Oh, I think maybe I do. Let's see. Yes. Okay. It's about, so I think it was the Dallas Zoo. Hold on. I was looking at all the poster presentations because that's obviously what I should do at the AZA conference. Yes, the Dallas Zoo conducted studies, and I didn't read the entire study. I read the poster, but I do have a link to the actual study, so I'll like look further into it. But basically what they did is they exposed visitors to what they called positive animal interaction, which was basically hugging goats. That was their scientific way of saying hugging goats. So they had randomized individual hug goats, and then throughout the day paid attention to if they donated to conservation, if they said they were willing to make changes about saving the environment in their personal life. They found that overall, it made them more receptive.
Huh.
So basically,
Priming.
right? Yeah, you're priming them with oxytocin is what you're doing.
Yeah.
Which we've done a lot of. We've talked about a lot of different research based on oxytocin priming. So they did what they called this, exposure to the goats. So basically hugging goats, and then whether they were willing to pledge to support some cause or make some behavior change, and then giving money. And it was statistically significant. They gave more money. They pledged to change things more often. I thought it was fascinating. And it's such a good reminder when I know anecdotally and deep down inside that zoos do positive work for the environment, for conservation, for science, and for endangered species. But it's always nice to see the data, I guess, because I am a scientist at heart, right? So when they do studies on people's understanding of environments and ecosystem services and all these kinds of things based on having visited a zoo or an aquarium, and that positive result is a great thing to see. Also, at this conference, I was getting trained to help lead my own regional climate change training in the spring, which I'm really excited for. And it's through this thing that I've talked about a million times on the show, NOCI, the National Network for Ocean and Climate Change Interpretation. So that's the big thing that I went through that lasted like 3 or 4 months, and I flew back to Boston a bunch of times. I'm sure you remember that. So back when I did that, I became this, certified through this process. And since then, in very intense evaluation, now by testing visitors at zoos and aquariums, they can identify the zoos and aquariums that have had management staff go through the NOCI training.
Oh, that's interesting. Well,
That's
yeah,
huge.
it
So
just changes the way that the zoo itself communicates.
right, and it changes the way they train their part-time staff too. It changes everything about how they talk about conservation issues, and particularly climate change. So the fact that you can poll visitors and know whether 2 people at an entire institution have gone through this specialized training, that's awesome. That's real change, because you hope that you're imparting this information to somebody who trains people, who teaches people, and it's kind of this upside down pyramid, right? Kind of this fan out of information. You hope that that's going to happen, but you don't know if trickle down ever actually works, right? Until you test it, depending on the subject. So in this case, it totally worked. So knowing that, and then in this case, just the interaction, the physical interaction of being able to be up close with animals, and then that primes you chemically to be more
relaxed, because you're like touching the goats and you're having nice happy goat time, and you're like, these are awesome goats, and then the oxytocin is there, and you've also got the opioid system relaxing you,
and it's kind of an empathy primer too, right? So it gives you the ability to be able to picture why these things are important despite our very busy, kind of removed lives that we sometimes have. that's why these places still exist, and that was really meaningful. And then the last thing that I was talking to you a little bit about right before the show, which there were a bunch of stories this week in the news about it, but I couldn't get into it on the show. It would be like a half an hour conversation, so I just didn't even want to get into it. But that the Threatened Species Commissioner in Australia, he spoke at this conference at one of the general sessions about the fact that I think it's like 30% of their land mammals are endangered, and it's all happened since European humans came to Australia, and it's because of feral cats.
Cats.
Oh, yeah. This is a global problem once again, people.
Yeah, and it's
Cats
because
are destroying the world.
there has never been a felid on the continent of Australia until we brought feral cats. And they go out into the bush of Australia where there are no humans. And, for example, one 6-kilogram cat that they caught had a 5.3-kilogram wallaroo in its belly.
What?
Yeah.
That cat had major goals.
So they're very effective at what they do, cats. And so if they're put into an environment where animals aren't afraid of cats because they've never seen a cat, that's a problem. This is the thing that's really cool, though, is that this Threatened Species Commissioner helped raise funding and supported the development of this new technology. He calls it, what did he call it? The cat paintball box. So it's this box that has a sensor on it. And I don't know exactly.
Cats with paintballs?
Kind of. Hold on. So it's like a. I don't know what kind of sensors they use. I'm assuming it's a camera that can analyze shapes of things. But if you're a bilby and you hop by, it does nothing. If you're a numbat and you wander by, it does nothing. If you're a kangaroo and you wander by, it does nothing. If you're a dog and you wander by, it does nothing. If you're a human and you wander by, it does nothing. But if you're a cat and you wander by, it shoots this little pellet at you that's like a paint gun pellet, and it doesn't hurt them right away, but it has, I think it has it's an anti-venom that I think has a neurotoxin in it. And but it was developed from this anti-venom, and so when the cats groom themselves later, it just slows down their heartbeat and they die. And
so this is really
hold on, and they want to develop it so that there's an additional element that is a microchip scanner, so that if you have a domestic cat in the area, because right now it's only in the wilderness, but they want to be able to put them just outside cities. And you're supposed to keep your cats inside in Australia because this is a problem. And so then if the cat wanders by and they read the microchip, it doesn't shoot the deadly paintball, but it does send information back, and that house gets a citation.
Oh, that's great. So I'm very terrified about an autonomous robot shooting neurotoxins.
It's not an autonomous robot.
It is. It's an automated system. It's an automated system firing neurotoxin at things that go by. I don't like that idea at all. I do like the idea of, cats are an invasive species. They're much like the cane toad is to Australia.
Yeah, cane toads are a big deal.
Yeah, and because of this sort of protected status of domesticated animal, because we like them and we don't like cane toads, they're viewed completely differently. But if it's an invasive species that's doing more damage than humans, if it does the same damage as humans, I guess we shouldn't complain too much. But we do a lot of damage, so we should get rid of even things that do less damage than us. But there've been islands, I think, around there. I don't know if New Zealand was the one that lost a lot of its flightless birds because of cats. Mm-hmm. It might have been rats that came aboard ship for that got offloaded.
New Zealand also has a massive feral cat problem with their endangered species.
Yeah. Mm-hmm. Yeah, so these are places where cats weren't. So everything that evolved there evolved without them. So yes, Fada, Homo sapiens are an invasive species, but we have an affinity towards them because we are them. We are people. So we are conquerors of the earth. We admit it. Yay, we won. But I don't think I don't think it's sufficient to give cats that same protected status in places where they're devastating to the environment. And if you've got your cat lovers who will put up feral cat shelters here and around Davis, and then you have your aviary fans who lobby against them because they want more birds in the backyards and want less cats out there. So this is
there's other
nature
ideas. It's well, okay. So in Tasmania, which is an island, they have officially collectively decided, okay, no more cats. We'll see if it
Cats
lasts.
have caused such a huge problem on Tasmania.
Yeah.
The cats have caused a huge problem there.
Yeah. So they've decided no more cats. People who have cats, as long as they're neutered or spayed, are allowed to keep them until their end times. But otherwise, you can't. They're not. Nobody's supposed to be getting more cats. In Tasmania.
Let's agree to get rid of all the cats. Look, we have documented sufficiently behavior to entertain anybody for several lifetimes on the interwebs now. We no longer need more cats. We've documented them. Their role in society is secure in history, but it's maybe time to move on. Can we find a different pet to replace cats? What would you suggest, Blair, as a good
dogs?
No, not dogs. Dogs are terrible. Dog owners are the worst people too, because they take their. You missed this. This is my rant last week about how they take their dogs to the park and let them run up around kids and stuff as though
you can't have a pet if you can't take care. If you can't be responsible, then you can have nothing.
And they aren't. People who have dogs aren't.
Then you can have nothing. The one thing you could have, I think, is a goat. You could have a goat.
We should all have goats instead of cats and dogs.
So another interesting study this week that we didn't talk about. There's a new species of tool-using crow that's been discovered.
What?
It's the Hawaiian crow.
Hawaiian crow. That's it.
But it's extinct in the wild due to predators. Should be like cats and rats and things, and also human use of land and stuff. But
strengths, llamas. Yeah, llamas. We could all have llamas. That'd be cool.
Hawaiian crows using tools. They're only in zoos now, though.
Well, if they've only shown it in captivity, it's hard to.
Yeah.
I don't know if it's hard to, on account of having seen it in the wild across the species elsewhere. I think
species or subspecies? That's the question.
This is a species.
It's a species. Yeah.
But this is, and so this is also. I think, especially just listening to this show, we can assume that all crows anywhere. Crows, ravens, what are they called? Canidids?
Corvidae.
Corvids. That the corvids everywhere are.
With a V.
Never mind. That these are really smart birds and that they're going to have this sort of behavior wherever they go.
Yeah, and it could be, maybe not all of them have the behavior, but whether or not it's shown in captivity shows that there is some proclivity for the learning of the behavior at a certain point. So it's whether or not it appeared de novo in the crow's mind.
Right.
I need a tool, and I will use it.
Oh, pigs. I would love to have little pot-bellied pig. That would be
my God.
Cool.
Strengths. Sugar gliders are one of the worst pets you can ever have.
Don't
you dare say it again.
Yeah.
What's a sugar glider?
Pet. They're super cute.
Oh, they're super cute. Pigs, aren't they intelligent? They're smart. What's a sugar glider?
Here, I'll show you.
They are a type of possum.
They're cute. They have big eyes and they are cute.
They're not what you're picturing, Justin. Those are sugar.
No, I'm talking about. I said pot-bellied pigs is what I said.
Look at this. They got that.
No, but strength is sugar gliders. And it is the worst idea I've ever had. So or have ever heard. So the sugar gliders have pointed, sharp, outward sticking incisors. basically like they have a dagger on the top and bottom of their mouth, and they use it
Aw.
to dig a hole in through tree bark to get the sap, which is why they're called sugar gliders, right? But that also means they bite so hard they will bite through a raptor glove.
Wow.
Wow, that's a big good bite.
How do I know that?
Because you've been bitten.
Yeah, it's terrible. And people have them as pets, and I see them like perched on people's shoulders, like licking their ear, and it just, I can't handle. Look at the little incisors on that middle. Yeah, there you go.
Those sharp little teeth. Look at this. A little buck-toothed little sugar
They
glider.
also smell terrible and are very loud at night. Twit refugee, how dare you? He says, animals just don't like you, Blair Bass. That's not true.
How many animals have you been bitten by?
I don't know, but I've worked with over 200 species. That's not counting individuals. So I would say I've probably only been bit species-wise by like 5% of the animal species I've worked with.
Which would be then 10.
Yeah,
species.
probably.
About.
Probably.
I got bit when I went to the visit you at the zoo.
Oh, by the
Got bit by an
ostrich.
An
That's
ostrich
not
bit me.
biting. You got gummed. If they really wanted to hurt you, they could have.
I didn't really realize at the time that ostriches don't have teeth. So I was.
Birds don't have teeth, Justin. Come on.
No, I know, but he was snapping, and then when he got a hold of my finger, I like screamed, ah! Like, ah! And then realized, oh, that really didn't hurt at all. But he was very snappy.
She.
She.
Mm-hmm.
Shishi.
Yeah. She.
Don't be so shishi.
Huh.
Facts. The trick is to bite them first. Yeah, that's how they know.
Yeah, I would say if you don't want to have a dog, get a goat. That would be my
Or don't have an animal.
yes. I would. Yeah, that's my first choice. Is just don't get anything.
I seem to have riled Dave Friedel. He wants to pay money to see an ostrich beat me up because of my cat comments.
Ow.
Emus are not a great pet. Strengths, you have the worst pet ideas. Just stop it. Oh my gosh. So Kiki,
Dar has been assaulted by a giraffe.
Yeah, so speaking of giraffes.
I nearly was. Actually, that's the other thing. Maybe animals don't like me. That giraffe was really sweet to my kids, but kept trying to smack me with its neck and its head.
Are you talking about the male?
The one that we were feeding bananas to. Just kept swinging
Justin,
its
this
head
was
at me.
like 5 years ago. I don't remember.
What were you going to ask me? You were going to ask me something.
Fishbowl Blair. Every 5 years, it's a full reset on your memory.
I don't remember which giraffe you were.
I didn't ask you which one.
I didn't ask
you which giraffe. It was trying to get me.
Kiki, I was gonna make a response to what you said in the chat room right before we finished talking about giraffes earlier, which is related to what male giraffes do to find out if it's time to mate with a female.
Yes, my link. Yes.
Yeah, so
that I shared in the chat room.
I would disagree.
What female giraffes do, which I don't know if we've talked about it before or not, but
I
might disagree.
We have. It's all hormones.
Yeah, but basically if a male wants to find out whether or not a female is in heat, they go through what's called the Flehmen process. And so the male licks the female's rear parts, and then the female pees on his mouth.
Mm-hmm.
And then he tastes the pee and scents the pee, because we know that taste and smell are intermingled. And he'll Flemin, you go. All right, Flemin response and determines whether or not the hormones are present for the female to be in heat and receptive for childbearing. And I didn't realize. Yeah, I've found. Yep, female giraffes pee in the male giraffe's mouth.
Yeah. So that's true for a lot of hoofed mammals, but also a lot of felids, a lot of cats. So when I was a zookeeper and I worked with the snow leopards, I had to observe the snow leopards because you never want to put snow leopards together who are not ready to be together because they're extremely solitary. You don't want to let them in together if the female is not in heat because she will tear him apart otherwise. So I would have to make observations all day on how many times I would see the Flemin response from the male, which would mean that the female would mark her territory, he would get a whiff of it, and then he would go.
The Flemin.
Flemin. And then I would make... I would also
it's like the wine tasters.
Because they're
I've seen wine tasters do that.
Yeah. Oh, it's the same. It's so it actually is supposed to improve the vomeronasal organ.
Mm-hmm.
So,
the vomeronasal organ.
Located above the roof of the mouth via a duct which exits just behind the front teeth.
Yes, and the vent, it's usually used for reproductive purposes, but the vomeronasal organ is known for pheromone detecting in mammals. And there's a question as to whether or not people have one somewhere in here still, because we can sense, but can we really sense them the same way that other mammals do?
And do we assume that we sense them or don't sense them in the way that animals do? Because maybe we do, but maybe like the animals might not know that they're sensing it, but their brain then just kicks into, oh, here's what happens now, right? Like maybe our brain just clicks into here's what happens now, unbeknownst to us, because the brain is doing things on our behalf or pushing us towards or away from things constantly without our conscious knowledge of it anyhow.
Now I'm just giving you turtles eating a flower. Baby
oh my
turtle.
gosh!
Baby sea turtle or baby tortoises. There we go. Tortoise hatchling.
Little Russian tortoises.
Little baby tortoises eating a hibiscus flower. That's pretty cute. Aren't they cute? My goodness. Yum, 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 chomp, chomp, chomp. Yum, yum. Chomp, chomp, chomp.
Yeah, Bex, brains don't always care what you want. This is true. It is. It's an abusive relationship, but it's an evolutionary hierarchy. You're not supposed to be in charge of your brain. This is a major hack job. This whole human intelligence thing. We hacked the brain and can seek out different stimuli as we choose consciously, despite what the brain might be trying to get us to do, to some degree. But on the other hand, the brain still has this hierarchical control over things like hunger, sleep, sex, reward systems, to an extent. But we hack it all the time.
Hack it. All right, you guys, it's 10 o'clock. I need to go because I still have to work on my presentation tonight and get some sleep and do the things that I'm supposed to do.
Okay.
Tomorrow is going to be a day of decongestants and coffee. I see it already. It's going to be one heck of a presentation. Here we go.
And nobody buy any exotic pets while we're gone.
Yeah, no buying ocelots or sea
capybaras.
turtles or capybaras. Yeah, no ROUSs. Just don't do that. You don't need that. You guys, thanks again for a great show. Thanks everyone in the chat room for hanging
Thank you, minions.
Thank you, minions.
We love you more.
I hope you all have a wonderful week. And we'll be back again next week, just like we usually are. I can't think of anything else in the intervening time.
Nope.
Yeah. Hopefully next week, the cold will all be gone. It'll all be good.
Yeah, I'm sure.
We
it
all
will be.
rest, and I'll be all rested and happy. And I'll be like, ta-da! But I did get my hair did.
All right, say goodnight. Say goodnight, Kiki.
Good night, Kiki.
Say goodnight, Blair.
Good night, Blair.
Say good night, Robo Justin.
Good night, Robot Justin.
Perfect. Good night.